Now, let me just quickly introduce Dr. Jana. So he did his BSc from Calcutta University in 2003 and MSc in chemistry from IIT Kanpur in 2005. Then he did his PhD from IISc Bangalore in the group of Professor Biman Jana. He completed his PhD in 2010. And then subsequently, he joined the group of Professor Jose Onuchik for his postdoctoral research. His postdoctoral stint was during 2011 to 2013. He was both at UCSD and Rice University. And then he returned to India and joined IACS Kolkata. And since 2017, he has been an associate professor in the School of Chemical Sciences. So his general research interest is in the area of theoretical and computational biophysical chemistry, and with particular focus on the mechanochemical cycle of molecular motor proteins and ice growth modulation by ice binding proteins. I guess today we are going to hear about this second topic. And he has received the very prestigious INSA Young Scientist Medal in 2018 for his contributions in this general area of antifreeze proteins and motor proteins. So with that introduction, I request Biman to deliver his lecture. And I would recommend that the students particularly should ask questions, and you can interrupt and ask questions in between. And yeah, it should be more interactive. So, and also if you cannot ask it yourself, you can put it in the chat box so that I can communicate it to Biman. Okay, so Biman, the stage is all yours, and you can start.
Okay, so thank you, Suman and Ranjitda, for inviting me. And so what I'm going to today is that I'm going to talk about little bit about antifreeze protein, as Suman really told here. Let me first present. Okay. Is it visible?
Yeah, maybe you have to turn on that laser pointer.
Yeah, that I'm doing. Okay, so let us first start. Okay, so what I'm going to do today is that I'll be talking a little bit about antifreeze proteins, and not really all the thing we are doing, but little bit off topic, that I'll try to connect two phenomena. One is called cold denaturation and antifreeze proteins. Okay, so I'll make it clear what I mean by this. But let's first give an overview of my lab. What we do is that statistical mechanical theory and mathematical and computational modeling of biological systems. We do work on motor proteins and some of the genetic circuit stuff. Then, of course, protein folding, antifreeze protein, amyloid aggregation, all this stuff. Okay, so let's now get into the topic. So, as the topic suggested, it is cold denaturation and antifreeze protein. I'll give a brief overview of cold denaturation first. Okay, what happened that proteins, when it is in the native state, by that what we mean actually, this is the native state. That means the state which has a particular structure and that is active biologically. But if you see this diagram, this is delta G of native to denaturation. Denaturation is a state. If it is denaturated, that means we say that it can lose its unique three-dimensional structure as well as probably its activity also. Now, proteins, generally we know that it is active within a temperature range. That means it is native within a temperature range. If you heat, then after a certain temperature, it gets denaturated. That means it loses its structure and also activity, and that particular point we call it's a heat denaturation of the protein. But what is even more interesting, if I go on decreasing the temperature, there also comes a point after which it loses its activity and to some extent also structure, and it goes to a state called cold denatured state, or the phenomena is called cold denaturation. This is the stability in which the protein, the green one, actually works. So now, why cold denaturation is, first of all, it is very poorly understood. That's simply because if you really go and decrease the temperature, what happens that cold denaturation and also the ice formation really makes it difficult to study protein at that temperature. And it has certain application also, implication in ecology and pharmaceutical industry. People have done some work, of course, and mostly I'll be talking about computational work, where they have studied this cold denaturation phenomena. As you can understand, it is very difficult because when you go to a cold temperature, then the dynamics becomes slow, and then this kind of seeing denaturation becomes really difficult computationally. But nevertheless, what people have found out that upon cold denaturation, the core hydrophobic moiety becomes solvated. That means these are the water, you can see yellow, and this is the valine, which is a hydrophobic residue, and that gets denaturated, I mean, hydrated as you go from 300 to 278 Kelvin. People have also studied beta hairpin, where there is a clear cluster of hydrophobic residues at the native state, but when you make it cold denatured, you can find that the hydrophobic interactions of the core is disturbed and water gets inside. Okay, we will keep this mind that this is a weakening of hydrophobic interaction that give rise to probably cold denaturation. Okay, now the second thing is called antifreeze proteins. Now, what is that? This is an interesting protein. I'll start with telling that if you think about Arctic region, the temperature is really cold there, and normal species cannot survive. That's because inside our body fluid there are lots of water, and it will form ice, and that means ice also has this peculiar property that when it goes from water to ice, the volume increases, and there is a certain phenomena called cell burst or ice burst will happen, and probably species will cease to exist. But there are certain insects, fishes, plants, fungi, soil bacteria, everything actually survives there. The question is, how do they do so? What people found out, there is some class of proteins which has diverse structures they produce, and these are the proteins which, what it does, it actually uh, makes something there so that the ice cannot really grow or ice cannot really form too big a cluster inside your body fluid, and that actually, that's how the body fluid still becomes liquid like up to a certain temperature, and they can survive. Okay, so those class of protein are called antifreeze. The name suggests antifreeze. Okay, so now the question comes that how do they do so? So if you think about it, how it works. So first, this is the classical nucleation picture. You know that beyond a certain critical nucleus, there will be unrestricted growth. This is the critical nucleus. If I think at a particular temperature, if you can somehow modify the growth kinetics and form the crystal or radius of the growing surface fall off or fall little short of this critical nucleus, I can actually stop the growth of the ice, and that's how antifreeze protein works. I'll tell you how. Let's say this ice front is growing. In your solution you have water, other proteins, and this is the AP red one. What they do, as you can see, they actually bind specifically on the ice surface, and when they bind, then this situation occurs. Ice cannot grow where it is bound, but ice can grow in between. That means you are certainly reducing the growing, the radius of the growing cluster, and if you deposit more and more such AP, then this radius will become even smaller, and at a certain point of time this will fall below the critical nucleus, and then your ice will no longer grow. This kind of effect is called Kelvin or Gibbs-Thompson effect, and that is how it works. But you can understand one thing, it has to actually bind to the ice surface. And then how do they bind it? So there is a long story about how do they bind and what are the debates and everything. I'm not going into go to that detail, but I'll just say one thing that from our lab we have proposed a mechanism of ice binding by host of antifreeze proteins of different structures. What they do, they actually do not bind to the ice structure itself by hydrogen bonding or something. What they do initially, at low temperature, there is a particular phase which is called ice binding site, where they actually form water ordering or clathrate kind of ordering, and that actually interact first with the ice and absorb on the ice surface. And if you go a little bit below that temperature, that particular clathrate structure actually becomes part of the ice structure, and the antifreeze protein is now quite irreversibly bound. That is what is the common mechanism across all the AFPs, which is clathrate dependent ice binding. The term you need to really remember from here that it has to create a clathrate like structure or anchored clathrate like structure around its ice binding surface. This will be important later on. Okay, having said that, I gave a little bit of overview of both the topics. Then what we will concentrate today is only the helical kind of antifreeze proteins. These are the two helical one is called winter founder AP, and this is a Maxi protein. All are helical antifreeze protein. And for cold denaturation stuff, we'll talk about TRPK's mini protein, which is also helical, and this is the model protein people work on. So let's think that what I am really talking about now. Let's get into the subject. So before I talk about all this stuff, let's first think little bit about folding of a protein. Or so for folding, people nowadays understand in terms of a funnel. That means a folding funnel, where this is the native state, which is the unique state of the protein where it has to fold to become active or functional. But of course, in the landscape, there are structures which are not so folded. And what by folding means generally not folded or unfolded basin kind of proteins actually through a path it guides towards the minimum of the funnel to give you the folded state. How do they really do in real system? Actually, people have found that initially you have a chain which is quite unfolded. You have these blue dots are water, and then it fluctuates, and suddenly after some point of time it becomes intermediate, which is kind of near native, but they have solvated hydrophobic core. That means some water is still there, and the most important part then happen at the last is the expulsion of water from the hydrophobic core to give you the folded state. What is the consequence? Consequence is simple. You have most protein, or most of the protein, if you find in PDB or protein data bank, you will find they have dry hydrophobic core. That means they are devoid of, mostly devoid of water. Okay, so then we go to an interesting topic here. From here, we'll try to connect things. Okay, first, so people are finding different structure of antifreeze proteins and finding how do they work and stuff like that. And suddenly in 2014, people found out an interesting antifreeze protein. They call it Maxi protein, the name. This is something like 4 helix bundle. That means the 4 helix forming a bundle. But what is even more interesting, that the core of this particular protein has lots of water in it. As I told you, the classic theory of protein folding or concept of protein folding says it has to have a dry core. But here we find lots of water. And not only that, most important fact is that this water has certain kind of structure. I'll tell what kind of, because I have already told the clathrate name. It has a clathrate kind of structure, and that is very important for this to bind to ice, because as you can understand, if it cannot bind to ice, then it cannot be active. So two things you get from here. First, a contradiction that there are lots of water. Why so? And second, those water are structured. I'll go one by one. But before I go to this particular structure, as I told you that this kind of cold denaturation I told you earlier, I have not connected yet, but that is dependent on the weakening of hydrophobic interaction. So let's think about little bit more general, forget about everything, little bit become more general and think about what happened to the low temperature hydrophobic hydration. It's more like a physical chemistry problem. I would try to think that how it works. Okay, so how to do that? Classic way of doing, you do potential of mean force calculations, bringing two methane molecule slowly towards each other and try to find that what are the minimas in the potential of mean force you are getting. Okay, so what you get generally at high temperature, there is a contact minima. Of course, two are hydrophobic methane, they will come close to each other and they form a two methane kind of thing and water surrounded by it. We call it contact minima. Then, of course, you find a first solvent separated minima. That means there are two methane with one layer of water in between. But what is interesting is that as we are interested in cold part, we actually went on to very low temperature. And what we find, there is an emergence of a second minima. And that minima is kind of simple enough. We have first solvent separated minima, which is only one water layer. Here you have two. Nothing so different, but you have two layer of water. But this will be important later on. You will see. Okay. So now let's think that first try to do little bit of this, that why this is coming, this kind of minima is coming. For that we have done some finite difference of the free energy or PMF, and I will not spend too much time. What I'll want to say is the contact minima that we know it is because of entropy, because when you take two methane together, then the water from the inside goes to the bulk and gains entropy. That's why you get stability of that minima. But both solvent separated minima first and second, that happens because of the enthalpy term. This is a 300 and this is a 240. You can see the enthalpy term is quite dominating. The question is that it is enthalpically favored, solvent separated minima. We can go a little bit further and see that because of what term of entropy, enthalpy it is giving you the minima. That means we can have enthalpy due to methane and solvent, due to solvent and due to PV work itself. And what we find finally, that this is happening because of the solvent only. That means at low temperature, it's the water-water interactions which is actually favoring to form the second solvent separated minima. That's what is our observation. And then we thought, okay, that's fine. This is 2 methane, too small, a hydrophobe. We can go a little bit bigger. We prepared a cyclobutane kind of stuff, or you can say square, having 4 methane in a square, and try to bring the same, play the same game. You do find 3 different minimas, but now find something interesting. There you are seeing that in the 2 methane, both the first and second solvent separated minima was stabilizing as you decrease the temperature. But here, first time, when you in little bit bigger hydrophobe, what you find, at you decrease the temperature, actually second solvent separated minima becomes little bit more stable than the first one. This is interesting. Then we thought, okay, why not just go to a rod-like thing? That means we put 10 methane one after other to form a rod and try to do the same game, that coming closer, closer and closer. And you simply find the same thing as I told you, that the second solvent separated minima becomes the stable minima at the low temperature. So at that time, this was the first time we were working on this, and we said that, well, the stabilization of contact minima and stabilization of solvent separated minima was responsible for denaturation of the protein. People used to talk about this, and we are just adding up to this that because protein has large hydrophobic moieties, not like only alanine, it has aliphatic leucine, isoleucine, phenylalanine, tryptophan, which are bigger hydrophobes. So that means we are telling this cold denaturation may have some role. That means the second solvent separated minima, not the first one only, has some role in doing the cold denaturation, maybe. Okay. So this is the first thing I wanted to say. Now go to the actual problem I talked about. What I said, that core has lots of water. Now you can try to simply understand where I am going there. You find that at low temperature, actually protein goes to a form where there are lots of water inside, or at least some layer of water inside at the low temperature. So can we connect this? Let's see. So that means there are lots of water. I will first try to understand the structure, nothing else. So this I am showing here because this is like a 4-helix bundle. In nature, there are other 4-helix bundle like rope dimer or something, which is completely dry core actually. So then why this is happening here? First thing we tried with the Maxi protein is to heat it up. That means run a simulation at 300 or 260 Kelvin and try to see whether this structure with lots of water is stable or not. We find no, it is not. It actually goes to a contact minima kind of state. That means two helices actually collapses and water expulsion happens from the core. That means this protein is not that stable at higher temperature. But if you do the simulation at 240, okay, 240, I should mention that we are doing in a water model where the TM or freezing temperature is not so high as in natural condition. It is around 232K or something. That's why 240K is reasonable to do. And what we find that this structure is actually stable. So we thought, okay, let's do a free energy kind of simulation and try to think about energetically what is happening. What we find is interesting, that at low temperature, we find the biologically active state or whatever the crystal structure states with lots of water is actually the stable state. But if you go to higher temperature, you find both the population, one with this kind of state, but other one is completely dry core. That means all the water is expelled out. Okay. Now, if I go to this state, both in the crystal structure and from our simulation, what I find, there are water structure inside, as I told you, but most important fact is that there are two layers of water inside this. And this is the part I wanted to talk about. It is not only that this helices forms a water core, but it is exactly having two layers of water. So now remember the hydrophobic hydration I talked about, that if you go to lower temperature, then you have a second solvent separated minima between the hydrophobic with two layers of water, and you are exactly finding the same thing here. That tells us something, that probably this is a state which is like a cold denatured state, a low temperature hydrophobic hydration kind of state, which is making the cold denaturations. But we thought, why not? I mean, these are all model structure. Why not prepare another model? Because this is a 4-helix bundle. So we thought that let's prepare something which resembles the structurally similar to this. That means these 2 helix and these 2 helix bundle, they are connected. These two are connected and these two are connected. And what you can see at higher temperature, this is the contact minima. That means you should think this way. There, that is the stable state. But if you go to lower temperature, that state is no longer stable, but only the solvent separated state with two layers of water is the stable state. So taken together everything, what we thought is that the Maxi protein structure, which is biologically active, is basically topologically like a cold denatured state with weakened hydrophobic interaction by inserting water molecule, what happens actually for cold denaturation. And for example, I'm showing here this slide I showed earlier, that at low temperature you actually do see the hydrophobic cluster is getting away from each other by insertion of water molecule. So what is interesting about it is that for normal protein, cold denaturation gives you loss of activity. But for this protein, I'm telling or giving you, or I'm arguing, let's say, that this is so designed that its structure at low temperature is like a cold denatured state, but that is what you need for this protein to be active. And what is even more intriguing is that because it's an antifreeze protein, it actually has to work at cold temperature. So maybe that's the nature's design. Okay, that's the first hint I'm talking about. Now think about something. I have water. So what? This water has to have a particular structure, as I told you, because I told you that this kind of water has to have a structure which is clathrate like, which I'll tell you later on, so that it can bind to ice. If it is not clathrate, even if it has lots of water, it doesn't make any sense because it will not bind to ice, so it will not be antifreeze. But it is here simply giving you this water molecule has a particular clathrate like structure. The question is how this kind of water is giving you a clathrate like structure. Now, the first thing I should tell you that why I'm asking this question. Whoever work on clathrate hydrate on stuff like that, it actually forms with entrapment of a gas and water forming a clathrate kind of structure. For that, you need a really high pressure situation. Otherwise, you will not find that kind of structure. But as you can see, this is a simple protein and it works in at least maybe low temperature, but not so low, but in ambient pressure condition. But you still find a clathrate like scenario. So we actually went on and dig a little bit deeper to understand that what is the phenomena which is actually driving this kind of water structure formation. I will not again go to the whole detail that how we did it and what we did it, but I'll just tell you the essence of it. So, so clathrate water can be characterized with some water parameter. I'm not talking about here that, but you can understand this is a hexagonalized. The clathrate has some slightly different structures, but both are ordered, and there are some order parameter called F4 or something like that, which take water dihedral, water dimer dihedral into account and can differentiate between the two forms. With that, we first went on to see what is the water structure in the crystal structure of Maxi protein. In the paper, they have said it is clathrate-like, but we still went on and checked that how it looked like. And this is the thing I was talking about. Inside the water, you can see here, there are lots of pentagon, water forming lots of pentagon kind of structure, polypentagon kind of structure, and there is a trademark of water having a clathrate kind of structure. So if you go and see the water inside the Maxi protein, this is the network they have shown. There are lots of pentagon it is forming, and you can run that code. You can code that F4 parameter and run, and you will actually see that is clathrate-like. The question I told you that when clathrate hydrate forms at very high pressure, clathrate in Maxi protein forms at ambient condition. Question is why? So we did a lot of calculations there, but finally what I find, that's what I'm going to say. There are three factors are essentially important. One, you should have hydrophobic hydration. Now, where from that is coming? If you check this Maxi protein 4 helix and try to see what is the amino acid composition, you will find there are lots of alanine. There are lots of alanine, at least close to 40% amino acids are alanine. That means it is having lots of alanine with CH3 in the side chain, which is giving you a hydrophobic environment. That means you need hydrophobic environment. At the same time, you need actually a little bit of confinement. Where from you are getting this confinement? You are getting this confinement because of this 4 helix bundle structure. Water inside is actually confined, semi-confined, I would say. But another important thing, what you need is the backbone anchoring. What does that mean? We find this clathrate structure inside this core is only possible if the backbone of this helix can make hydrogen bonding to the water molecule inside. Some kind of anchoring effect. That means its structure is forming and that is anchored by the backbone. Okay, so these are the three factors. We said that if these three are present, absolutely, if you just miss one of them, classed structure is probably becoming, it will destroy or it will reduce the ordering or something like that. Okay, so with these three, you get this kind of structure. So we have a factors which are governing this kind of classed formation at ambient. I would not say ambient temperature because temperature is a little bit low, of course, but at least in ambient pressure condition. Okay, so up to this, then I said two things. One, at low temperature, you get second solvent separated minima, and that I have connected with a protein structure which is quite, or I would say, little bit surprising, because protein generally has hydrophobic core, but here this antifreeze protein actually do have water inside. And then we said that there are exactly two layers of water present, and in second solvent separated minima also there are two layers of water present. That means topologically, I said that it is probably like a cold denatured state. And then the water molecule has to have a claptrade like structure, which is they are actually, we know from the crystal structure and also from our simulation at low temperature. But then we go and see and talk about, let's say, what are the factors which are actually important for that, for those kind of claptrade kind of water formation. Okay, so last few minutes I'll talk about little bit deeper or little bit structural details. Let's say till now we have talked about some topological, that okay, there are two layers of water and things like that. Now let's directly connect these two. So important part will be this backbone anchoring. That means backbone has to interact with water. So first thing is that it is a helical protein, and what you know for sure, that to form a helix, there has to be a strong intra-hydrogen bond. That means backbone has to form intra-hydrogen. That means residue 1 will make hydrogen bond with residue 4. And helix really doesn't want to do too much of backbone water hydrogen bond kind of thing. It actually prefers to backbone hydrogen bonding. That we know. So then we thought, okay, what is happening here? Can we understand a little bit the structural detail? Then we went on and do a structural analysis of type 1 antifreeze protein, which is helical, of course, and try to make sense of it. And there comes the first surprise. What we find that in type 1 antifreeze or Maxi protein, as I told, it makes lots of backbone water kind of hydrogen bonding. But if you go to a standard helix, which we see in normal rope timer or some other helix, their backbone is compact. They don't want to really make backbone water hydrogen bonding. Then we see, okay, let's do some helix parameter. So what I find is that alpha helix here little bit swollen. What does that mean? In normal standard helix, there are 3.6 residue per turn. Okay, this we all know. But if you go to the Maxi protein kind of structure, then you find it is actually not 3.6, it is actually 3.7 residue per turn. Now, why this is important? Both of them, this of course work on cold temperature, this ambient protein generally, this is important for the ambient condition, not so cold condition. But why this thing is important? You can understand that in a turn, if there are 3.6 residue, normal helix, but if you want to put now 3.7, so you have to swallow it little bit. And for that adjustment, earlier the perfect 1,4 kind of hydrogen bonding in the helix now will be disrupted little, and probably that will allow your backbone to interact little bit with the water. Okay, but that is fine. That is the two structural things we are talking about. Now, the first question we ask, let's take a standard helix, a protein which is normal protein, and let's cool it to make it cold denatured, and try to see exactly this parameter, that alpha helix residue per turn, how it is changing with temperature. For that, we cannot do normal simulations because because at low temperature things are pretty slow. So to have this cold denaturation kind of simulation, Devaniditi and Qualcomm also has done earlier. We have tried something called REMD, replica exchange molecular dynamics. What happens that here you have same proteins simulated at many different temperatures, starting from low to very high temperature, and after some time we exchange with Metropolis or some kind of algorithm acceptance, we exchange the structural ensemble between different temperature, and then that's how we try to see that how different structural ensembles stable at a particular temperature, because as you can understand, whatever is happening at some particular temperature, it doesn't have only one structure, but it has structure from other replicas of higher and lower temperature, because we are mixing that. And then finally we get the once it is, let's say, have done enough number of sampling, then from the trajectory of a particular temperature, we'll get what are the structural ensemble possible for that kind of system. So we have taken this TRP case, which has a helical structure, of course, one part, and then it is small 20 residue, alpha helix clearly defined, first polygon doesn't matter, and reported data you have computational study, so that we can actually make sure that we are not doing something really funny here. Okay, so first thing what we find is this. Remember that I told you that it is 3.7 residue per ton which is there in antifreeze protein at working condition. Here at high temperature where this protein actually works, it has a standard helix which is 3.6 residue per ton. This is number of residue per ton calculated from REMD simulations trajectories with different temperature. So it is quite stable with 3.6 residue per ton, which is like, I would say, let's say native helix. That's how I will talk about. But then around 220, a little below that, maybe, or around 220, we'll find a sharp increase in the residue per ton, and it becomes clearly like 3.7. So that means there is a subtle helix to helix kind of transition. That means it's both are helix, but there are some differences between them. So the cold response of this normal protein is that it is getting swallowed and swallowed and actually becoming a 3.7 residue per ton helix. That is the structural thing we are finding for cold denaturations. Let's think more that why this is, I mean, what is the thermodynamics and how it is happening. Okay, first thing is that we also at very many different temperature we calculated what is the backbone water hydrogen bond kind of scenario, and as I told you that when you go to a little bit 3.7 kind of turn, it is swallowing and it makes more kind of backbone hydrogen water hydrogen bond, and that is actually, if you see 213 Kelvin, you can find that this red plot, it is shifted towards right. That means it is making more backbone water kind of hydrogen bonding. Okay. So that is fine. And we then calculated delta G, and we find that delta G of the protein is actually really decreasing. Delta G means it is denaturated to between the two helix we are measuring in a two-state model kind of scenario. One is helix with normal 3.6 ton, another is helix with 3.7 kind of term. So it is actually decreasing, like you have seen in the first plot, that it is getting unstable, and it started actually getting unstable around 230 only. And then what I find from this analysis, we found 220 Kelvin is the temperature where it is becoming denaturated, and reported previously 225. So we are more or less there. We went on to do little bit more, that whether it is enthalpic and entropic, and we found that it is associated with delta is becoming less. This tells you that this is an exothermic kind of situation. Entropy is not that favorable as T delta is becoming positive. So what we find here is interesting, that this is an exothermic process, and people really do know that cold denaturation is also an exothermic process. So there is a helix to helix transition is going on, And that is probably the trademark of the system becoming cold denaturated. That's what our point is. So we have temperature dependent free energy map also. If you go on decreasing the temperature from 290 Kelvin back to 210, you will find that initially it is 3.59 or 3.6 helix, then slowly it is exploring helix with more residue per ton. This is the residue per ton, and this is RMSD. You can see just the x-axis. So it is sampling two different states. One is 3.75 helix, another is 3.65 helix kind of. And there are population transfer from 3.959 to 3.7 helix or 7.5 helix. 7.5 and 6.5 making probably 3.7. And there is a refolding happens after 213 because 3.75 helix comes back to again 3.65 also. What is interesting, I'll just not spend time here, but people do find that there is a refolding occurs actually. So first there is an unfolding or denaturation, cold denaturation, and then there is a compactness happens after after a certain temperature also. So probably this is happening because of that, but I don't want to comment here on this. And we have now tried to see that why it is happening. Long story short, we find, you look at this one, 213 kind of thing. This is happening because of the collapse, but forget about that. This is happening because of water interactions. So that means water is playing a major role. So can we connect this cold denaturation to something related to bulk water? Yes, we do. So what we can do is that we know that if we plot the density of water with temperature, and this gives you a maxima, and there is a minima actually also it gives you. You can also plot what is called the derivative of that. So this is the density plot, and this is the derivative of the density, blue curve. So you find that that has a maxima derivative around 230 K. Okay, and at the same temperature also supercooled water gives you Cp or kappa T maxima there. So what we are trying to say is that we know water is peculiar. It gives you, I mean, we know that if we decrease the temperature, all this response function kappa T and Cp should just decrease, but it actually goes through a maxima. And that we know it is related to the density maxima or change in density with temperature, as I'm showing here. So that also happens in a very similar temperature like 230 K. And we know that this can also be related to something called high density, low density kind of liquid phase transition. So don't go into detail, but simply that we can calculate the ratio of HDL, NDL, and we find where the HDL is going to LDL kind of scenario. That means the change is maximum. That is where, or this is the derivative I have plotted. So that is where you find this kind of transition. So if I am now telling that this particular transition or cold denaturation transition is related to the liquid structural change of going from HDL to LDL or density maxima, it is related to all these things. And we are doing simulation. So that means if I change the water model, this particular transition should change, because depending on water model, these temperatures actually changes. So what we thought that we do same REMD simulation of the trip gauge or cold denaturated proteins, cold denaturation phenomena at a different water model, let's say SPC. That has a slightly lower temperature. So you understand, one is 230 K. Now this one has a slightly lower transition temperature, around 185 or something like that. Then what we expect that this helix turn increasing from 3.6 to 3.75 or 3.7 should actually also get delayed and go to the lower temperature. And what we find is exactly that. If you go to the SPC kind of model, the transition temperature becomes like 185. Okay, so that means this is actually the water playing the major role for this helix to helix transition, which is responsible for cold denaturation. Now, helix to helix transition of what? 3.6 is going to 3.7, right? And I told you earlier that Maxi protein or type 1 AP protein their structure is so designed that actually 3.7 helix is the stable state or working state. That means it is already swollen. It's like denatured state, cold denatured state. Now, if that's so, then if I increase or do a temperature dependent study of one of the helical antifreeze protein, I should also see the similar kind of thing. That means at high temperature, it should go to 3.6 kind of scenario. And it is true. What happened that if you take this kind of antifreeze protein, they also show a similar kind of transition around 230K. And if you go a little bit up above 230K, it is actually going towards 3.6 helix. Okay, so what we are trying to say is that because this kind of protein has to work on very low temperature and inhibit ice growth and for that it has to make hydrogen bonding to water to form a ordered water structure which can actually bind to ice. That's how nature designed this kind of protein in such a way that it becomes stable at 3.7, because this is a little bit swallow helix and that can interact with water, and essentially that promotes the ice binding. So then that is what we are trying to see. Normal protein stable here. If you decrease the temperature, it goes to 3.77 helix and it is not active. Antifreeze protein in normal temperature, it is again 3.6, not active. But if you go below certain temperature, it becomes 3.75 or 3.8 here, kind of swallow helix structure, and this is where it is active. And when it becomes swollen, I told you it can make more backbone water kind of hydrogen bond. And you can see this is one of the essential thing for clathrin kind of water formation. As I told you, the factors governing it is nano confinement, hydrophobicity, and the backbone water hydrogen bond kind of scenario. So taken everything together, both structural, having water inside, and that is also 2 layers, and then having a backbone structure which is like a cold denatured state. What we are trying to say is that cold denaturation is a phenomena, of course, which is classically defined that a protein natural state or native state gets disrupted, activity gets zero while you make that transition. This kind of protein, antifreeze protein, most important thing is that it has to work on the cold temperature. So their structure or rational design is such that it too forms a cold denatured state. And most important fact is that nature uses at its advantage, because in that kind of state it can make water around it structured so that it can bind to the ice and therefore make the Kelvin-Thomson effect comes into picture, and the ice growth is halted. That's what is what I wanted to say. This is this. So if you have a normal 3-point helix, the clathrin water is not that strong and it cannot make too many strong interaction with the ice, and that's why the binding will not be so good. But if you go to 3.7 kind of helix, it has enhanced backbone anchoring ability and therefore strong binding with the ice surface. Okay, so AP is in the cold denatured state in all practical purposes like structure, water structure, and everything, but it is active. That's what it is. So summary and conclusion: the pairwise hydrophobicity at cold temperature exhibits second solvent separated minima. That's what I told you earlier. Then second solvent separated minima can explain the unusual structure of antifreeze proteins with hydrated cores. And okay, supports expulsion, forget about it. And then presence of appropriate confinement, hydrophobic environment, suitably positioned hydrogen bonding anchoring groups governs the formation of clathrin. Now temperature dependent helix to helix transition may have implication in cold denaturation and also in antifreeze activity. Okay, so the work which I presented was done by Srideep, and some of them by Sandipon. And I should acknowledge DST, CRB for funding, and CRE computing facility for IACS also. And thank you everybody for your kind attention.
Okay, thanks Biman for a very nice, insightful talk. Now the floor is open for discussion. Questions? Okay, in the chat there is a question from Dibyendu. So he has asked, is F4 order parameter alone is enough to say if I is having clatrate structure or not? I mean, can there be different I structures sharing same F4 value?
Yeah, so this is a good question. I didn't go into that. So generally, we do Q6 or kind of bond orientation or a parameter to understand what a structure, where Q6 has some value, tells you whether it is ice-like or not. But what we go when we try to see both hexagonal ice and classified water, we find Q6 are very close. That means one cannot really separate with these two. And then we went on to literature and find that what people really think about the classified literature, and they also, of course, found this problem and come up with this F4. F4 is something like that: you take two water, which is forming like a dimer, and then take two farthest hydrogens, okay, and then OH, OO, and OH. This dihedral angle, if you take that as a phi, then 3 cos phi or something like that, I forgot that, if you can define order parameter, which gives you that even if the Q6 is very similar, for ice, the F4 becomes minus 0.3, for clatrate, it becomes plus 0.7. That means you can separate out two structures. You can distinguish whether it is clatrate or ice.
Okay, next question. Manoj has a question. Please go ahead.
Yeah. Hi, how are you? Good?
Yeah, good.
Yeah, so it was a very nice talk, yeah, and very clear. So I have kind of one query, not a question, like, have you like calculated any pressure effect? So what should be the effect on high pressure? Like
Yeah, I
in
mean,
your
see,
case,
I understand your point that high pressure also gives you a denaturation, right? Even if at the lower
yes, yes.
normal temperature, the pressure induced denaturation you are talking about. No, we did not, because we went to cold temperature simply because we are working on antifreeze proteins. And that's why we thought that we'll explore into that direction. Yes, I mean, high pressure phenomena, I don't know exactly what happens there. I mean, there are also people talk about some hydrophobic hydration is becoming a problem and things like that. And also it has to do with water density change with pressure. But no, we did not.
Okay, so because why I'm asking this one, because like you saw two solvation cell, yes, fast salvation and second salvation. Yes, second salvation is like energetically more favorable than first one, but it's not huge, kind of
No,
similar.
yes, you are right.
So what I was thinking, if you create more pressure, then like more water can release, and then it may go to the first kind of first salvation cell type structure, and once you can reach there, maybe it's easy to unfold or like this or no.
Yeah, possibilities are there, but of course we need to do to understand it. I mean, I cannot really say
Yeah.
this from here that what is going to happen. Yeah, but
Yeah,
it's a
very, very nice talk. Yeah.
yeah, good suggestion. Okay, thank you.
Any other question? Anybody else?
Biman,
Ranjit.
yeah, just a query. That is for the protein which are going this cold denaturation, the water which are there at the backbone of these proteins. Is there any study of what happens to their dynamics around that temperature? Have you found out?
I don't find many. I have a plan to do. I have not
Okay.
started yet, but no, not many. Actually, the problem is that studying this cold denaturation is a problem. Even, I mean, experimentally there is a lots of problems simply because the ice formation intervenes.
Right.
So that means you have to somehow stop the ice formation to
Right.
study the cold
Right.
denaturation. Computationally, mostly the work comes from Devenadetti's group. Uh,
Okay.
there are few works, and that is also very difficult because other than REMD or some kind of enhanced sampling kind of technique, you cannot really do anything here because things are really slow. But this is an so maybe you are talking about when this 3.6 to 3.75
Right.
jumps
Right. Right.
happens, whether there is a change in the dynamics.
Anything drastic, something like
Yeah, I
that, a
mean,
sharp
this
change.
because you have all the data, you can actually check it. This is a good
Okay.
suggestion, actually.
And another associated query is this, that these proteins survive the really cold temperature. For
Yes.
example, for bacteria
Mhm.
or the organisms which survive at that low temperature. What happens to the water dynamics at those temperatures? Whether the water is actually executing very heterogeneous dynamics,
Yeah,
the
I mean, I understand completely your point.
right,
Even
right,
you are thinking of doing this dynamics inside
right,
this Maxi protein. That means there
right,
are lots
right,
of water,
right,
at least 400 water molecules
right,
forming this kind of structure.
right, right,
But
right.
we never did it simply because nobody is doing it. So we need
No,
to do. Actually, it is an
yeah. My
interesting
question
point.
is, I'm trying to get at to the relation whether the remnant mobility of the water molecules are connected to the functionality of these proteins at those low temperatures.
I understand
What makes these proteins alive at that low temperature?
at that low temperature.
Right. Whether it is something very defining role of water dynamics through spatiotemporal heterogeneity
Yeah,
playing any role
I
or
get your
not.
point.
Yeah.
The Ranjit, the point is that so these are all very recent thing, and so we were more interested in the structure at first
Right. No,
because
no.
this
Definitely.
structure of the
Definitely.
protein is so peculiar.
Right. 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 Right.
But
Right.
we didn't really do dynamics at all. But
Okay.
I think it is probably a good idea to do the dynamics
Right.
also.
This interconnection.
This is
Interconnection. If there is
interconnection. Yeah,
something
I know. I
about the,
understand.
yeah. Yeah.
Okay, thank you. I will
Thank you.
do some for sure.
Okay, I have a few questions. So depending on, I have many questions, but maybe I'll ask a few. So first thing is that I was trying to understand that what is the design principle. So there are two aspects that you highlighted. When you say that it is in the cold denatured state, so one aspect is that at that temperature, it has that sequence, that spacing. So that is one structural aspect, that the protein structure, it should have an optimized structure. But there is also, you invoke the water, that there is this LDL, HDL conversion is going on. So is the protein changing, optimizing the structure of the protein itself, or the water structural change? So which is important? So what is being
that
utilized
that
here?
is
So
that is a that is I think a chicken egg problem again. The point is that it is, see, the issue is that when I'm telling it is mostly water playing this role, I am not really telling the full story simply because of the fact, yes, water is guiding because I change water model and so that water is guiding this transition. But at the same time, I did not really highlight, you will see that naturally cold denaturation happens at little bit lower temperature than the activity of the antifreeze proteins. That means antifreeze proteins itself makes this transition little earlier. That means what I'm trying to say, it is the protein sequence which also has a role to make this transition little bit high temperature, because it doesn't want to go to that cold temperature because it has to work little bit, because cold denaturation happens something really low. This one has to work around -5 or -10 degrees centigrade, right? This AFP. So it is both is having the role. The question I'm trying to say is that then one can ask that any cold denatured helix state should be antifreeze protein, right? But the question
yeah.
is no, answer is no, simply because that I'm not telling simply that cold denaturation and antifreeze proteins are the same. I'm telling structural and other topological signatures are very similar. Okay, but what is important is that cold denatured, antifreeze proteins are so designed that it becomes like cold denaturation, but
here the denatured state is the functional state. So
that
the
into
functional
advantage.
state is the
It
it
takes
is
that into
utilizing
advantage.
that.
Yes, that swallow and helix making backbone water hydrogen bond and forming a platelet kind of structures. So I also think something, I don't know, I'm not sure, that cold denaturation state can have fluctuation which is probably more than the antifreeze proteins. That means I'm telling that even if I'm talking about a single point agenda kind of thing, that 3.7 helix, but if I look into the dynamical aspect like Ranjit was asking and other things, then I'm sure I'm going to find some difference between these two ensembles. That means between cold denatured ensemble and antifreeze ensemble, and that might actually define where the missing link is.
Yeah, and the other question was that since you mentioned that first there is a formation of clathrin-like layer, then it binds to the ice structure. So, but as you also mentioned, the clathrin structure is different than the ice structure. So when it actually binds, there is a mismatch in the structure.
No, that is very
So
interesting.
how does it help? Or
I did not talk about this because that is completely on the recognition part. That story I did not tell. I just gave one slide. What happened that if you have a growing ice surface, on top of the growing ice surface, there is actually a clathrin kind of structure. The water,
even in normal water, in liquid water.
Normal water. So it's the ice, and that surface is rough, and then it has some effect till 2 to 3 layers. We are actually exploring that up to what distance it can inform that there is an AP and stuff like that. And then you see that structure. What is interesting, all this AP binds to specific ice planes. And they form the clathrin structures. They are not all the same. They have different clathrin structure for different class of AP. And we matched that structure with different ice plane clathrin structures, and we find wherever the matching, they actually bind there.
Okay. Okay. And maybe the last question is that for normal like methane hydrates, we know that those cages cannot form without the guest. So empty cages are very unstable. But in your case, since there is no host as such, that there is no gas molecule, so do you find empty cages or not inside that Maxi?
Now the point is here is no gas molecule is not a right statement.
I mean the side chains are there. I understand it will form cages around the side chain.
That.
But otherwise, do you see any kind of cages which might be just because there is a lot of water, right? So there will be clathrin around the side chains, but other than that,
no, other than that you won't find.
Okay.
No.
So it is always around the side chains.
It is always around the first 1 layer, 1.5 layer, something like that you will see.
Yeah.
So yeah.
Another maybe quick note. I mean, this is maybe I found it curious. Maybe that did people investigate the difference in solvation behavior between HDL and LDL water? So I'm talking about their dynamics or their like how they will interact with this backbone. Their thermodynamics, how it changes between HDL and LDL. So because that will affect the thermodynamics, the solution thermodynamics.
The point is that I am right now getting a little bit interested, but then lost touch because you know that I sometimes used to work on this, but then I am coming back again, I would say. I haven't seen much, but maybe there because I don't have really the full idea of
because many of the like solvation properties of water might be drastically different between these two types of water.
Yeah, there is a work by Peter Ham on the antifreeze proteins where he found that from the second layer there is an accumulation of LDL. They said that because the first layer they said that they cannot really characterize what kind of water it is, but then in the second layer they find in the ice binding surface there is an accumulation of LDL.
Okay. Interesting.
So people are doing some work.
Yeah.
Hopefully we will also do some.
Okay. Yeah. So Rajiv has a question. Please.
Hi, Biman. This is not a question. This is just a kind of information. In case you are interested in the dynamics of the antifreeze protein and the cold denaturation, so there had been some experimental studies, in fact. So there was a very seminal paper in PRL, which was published in, I think, 2008 by
I know that.
the Markles group, you know?
Yeah. I know.
Yeah, so that actually tells that the protein backbone is not actually required for any just kind of transition. So that was very interesting study. I do not know if there is any further study regarding.
No, but it has to. So I don't know exactly what kind of system they have taken, but is their system is a helical system?
No, actually they have taken some kind of. I mean, I say some 7 or 8 long. Chain of peptides. So it's not a kind of helical. So it's not. So actually, so they said that what the paper says is that the kind of helicity is not very important on the transition. So they say that irrespective of the helicity, so they have chosen some say 10 or 12 amino acid polymer, and they said that it is not the helicity that is important for the 200 Kelvin transition.
Yes. No.
So that was the experimental, finding what they have actually said.
Yeah.
I am not sure whether there has been any further studies on.
I'm not sure also, but what I'm trying to tell you here is that cold denaturation is a phenomena which is independent of what kind of protein you have. Generally, most protein goes to cold denaturations. Here, most cold denaturation I'm talking about tryptase, I took that simply because it is first of all well studied, and second thing is that it has a helix in it, and I want to connect to the structural aspects of helical antifreeze proteins. So that's why I took it. And for this stuff, I know for sure that this, nobody showed this 3.6 to 3.7 that detail at transition.
Yeah, that is not in
but
the experiment.
People have shown there is a change in alpha RMSD, so the ideal helix actually goes to a non-ideal kind of helix. That means there is a change in the helical parameters, but nobody really go and check that what is happening. We got interested so much because we have seen that in the structure of antifreeze proteins, there is a clear change. It is going to a 3.7 helix.
So there has been some also studies on the antifreeze protein. The Mishkabon group or the Wimbacher group, they are doing a very detailed studies on this antifreeze protein.
I know.
And so you can, just in case you are interested in that study, you can do this.
I am pretty much interested. If somebody wants to do some experiment, I'll be really delighted to talk. Okay, I'll email you. No problem.
Okay, thank you. Yeah, fine.
Is there any other question?
Okay, so another quick question. So when you have like 3.7 residue like per ton, so what is the change of hydrogen bond length? So
hydrogen bond length, okay, that we did not see, but we can think about it, because so what happens if you have 3.6, then you have a perfect 1,4 directional hydrogen bond. If you make it 3.7, I believe that the length of the backbone hydrogen bond will increase a bit. I'm not sure. It can happen two ways, because hydrogen bond can happen, weakening of hydrogen bond can happen by increasing the length, but it can also weaken by geometry. That means the angle criteria. So that means by swallowing, I don't know which one is getting affected. No, we did not do it. Yes.
So why I'm asking this question, because I want to know whether it's going to like from standard hydrogen bond to low barrier hydrogen bond kind of things. Or
yeah, that's what I'm exactly talking about. That whether it is going to a compromised kind of scenario. It is going to a compromised scenario for sure. That's why it is making other hydrogen bonds with water.
Yeah. Okay, thank you.
Anything else? Okay, if there are no other question or comment, let us thank today's speaker, Dr. Biman Jana, for a very nice talk and discussion. So thanks a lot. And we welcome you when things become more normal. We welcome you to visit.
Yeah, because I have to go to SNBOS and have lunch and stuff like that, right? That is much more important.
Yeah. Okay then.
Okay.
Bye bye.
Thanks everybody. Bye.
Thanks.
Bye.